What's up, guys? So in this video here, I'm going to be talking about mutations, and so we're going to be looking at the types of mutations there are. There's the point mutations, and then there are frame shift mutations. And within the point mutation, there's a few different kinds. This video also goes along with the Student Notes 9 on mutations, which has also been uploaded on the Teams, so feel free to take that out and go along with it, though I'm going to be going through some of the parts a little bit more quickly, just so that the video is not crazy long, and that I can talk about certain more complicated things a bit longer. Okay? So first of all, a gene mutation that I kind of mentioned in some of our Zoom classes before is that it's essentially when the sequence of the bases gets changed in some sort of way. So remember that there in DNA there is A for adenine, T, thymine, G, guanine, C, cytosine. And sometimes those can be changed for a number of different reasons. And if the overall sequence is changed, then that means the amino acid sequence would also change, and then thereby the protein that it's making would be affected. Okay? Proteins are quite dependent upon their shape for how they function. And so if any of the pieces in there get changed, there's a possibility then that protein will no longer function as intended. Okay? And the thing with that too is that if the DNA sequence itself mutates and changes, that means the mRNA that would be read from there is also going to be affected, which thereby will affect what happens at the ribosome. Okay, so there again, final protein is changed. Okay, so an example here is if you see here, there's the sequence CTT right up here. Okay, which would mean that the complement from the mRNA would be GAA, but what happens in a mutation is that for some reason one of them gets changed. There's a number of different reasons, and I'll quickly go over those. Might get changed, and then that will affect the complement right there, which then affects the resulting amino acid, which you can't see because of my face, but it would have been there. Okay, so some of the things that would cause a mutation is that sometimes there's just an error in the DNA replication. Okay, like I've mentioned before, it can happen. It is quite rare for it to happen, and honestly, sometimes the changes won't actually result in anything, which we'll see in a little bit. But sometimes when there is a new DNA strand being made, there's going to be a mismatched pair. Sometimes by accident, instead of putting an A with the T or a C with the G, they put in the wrong thing. So maybe, for example, the G is paired up with an A. So that could be an issue. But this isn't all that common because our body has built-in mechanisms to fix these errors when they do happen. So there's an enzyme in your body called DNA polymerase that is going to proofread any of the work done while replicating to make sure that there are no errors, and it's pretty good at its job. Okay, it makes a mistake, maybe misses something in about 1 in a billion bases copied. That's kind of impressive. You think about if you were to write, an 1,000-word essay without making a single misprint when you are typing, that's pretty good. I mean, it's probably longer, honestly, in terms of the actual number of key strokes that you're doing. But just thinking about that, without making a single error for an entire essay would be pretty impressive. Some other causes would be something that are called mutagens. So these could be chemicals, viruses, bacteria, maybe energy from radiation that can induce a change to the DNA sequence. Okay, so things like UV, or X-rays, or radioactive materials, they can cause a change at the DNA level, which is why we're typically concerned with those things, and why we say you want to avoid as much of those things as much as you can, or you want to try to limit your exposure to the X-rays. And so the technology there for radiologists has gotten a lot better. And same with UV. You wear sunscreen, or you want to stay out of the sun for extended periods of time, because in some cases it can lead to as you probably have heard, skin cancer. And there's other chemicals, pesticides, compounds in smoke or cigarettes that can affect the DNA at the base pair level. Okay? Then again, just like with the random errors in replication, the rate is still fairly low because, again, your body understands how important the DNA is. And so there are a number of different repair enzymes that are constantly going through checking them to repair the DNA. Again, it's not 100%, but it is pretty accurate and pretty good at doing this. Okay, so just mutagens there. Oh, there's a little. Essentially what happens is these radiation or the chemicals, they can damage the gene and then make it unreadable. So sometimes, like I mentioned though, these repair enzymes, they're still there to replace any damage or missing bases. So if it does go through and it sees that a G is incorrectly replaced or paired up with a T, it's going to go through and fix that and give the proper pairing there to avoid any issue. Okay, so just like with UV radiation, say you're exposed to some UV, what can happen is that it can very temporarily open up a strand of DNA, just cause it to break apart, because again, those things are held by bonds, and the energy can sometimes that's added to it can break them apart for a very, very brief period of time before it does get repaired. In that time though, if it's not fixed, sometimes they can do weird things. Okay, they want to be bonded with other things. That's why they are called base pairs, and so they want to go with something else. They don't like being on their own. So what can happen then is that they kind of bond with something else, or maybe something else rushes in to go in there and just satisfy that bond. A lot of different things can happen. Essentially though, the UV, X-ray, anything like that, any high radiation or high energy radiation or anything like that can temporarily break bonds in the DNA, which then gets affected in terms of what it is actually bonding with. Okay. Another weird one, but we won't really talk too much about this, is something called a transposon. And so essentially what happens is that a DNA sequence, most of your genes have a set location within a chromosome, and for some reason or another, it just kind of gets shifted when replicating. It just makes a mistake. And then it moves an entire sequence of a DNA somewhere else, and then it kind of blocks the effects of other genes after that. Though I'm not really going to go too much into it, but an example would be, here's your normal gene, and for whatever reason, the gene that codes for, the white kernel instead of the purple one, it just kind of jumps in there, and it gets red first, and it just kind of supplants it. Again, I'm not going to talk too much about it, but it just is a type to be aware of. Okay, so in terms of types of mutations, like I mentioned, there are two that we're going to look at, and one of them is the point mutation. Okay, and so a point mutation occurs when a certain base is replaced by another base. So for example, we may have UAC, okay, and then for whatever reason, the C gets replaced with a U, okay? Got mixed up. It does happen. It's not crazy uncommon, but at the same time, your body is good at replacing these things. So that's a point mutation. One base pair is replaced by another, and the results could actually be quite different depending on how what it's changed to. Okay, so the first type of point mutation that we'll look at is called a silent mutation. Okay, I wanted a highlighter. Okay, a silent mutation. So in this case, for UAC to UAU, if you actually look at our codon chart, remember they actually code for a certain amino acid. So UAC, if we were to follow through, UAC would code for tyrosine. Okay, great. If we get changed to UAU by mistake, if you do look at your chart and follow through all the way through, it still ends up being tyrosine. So the amino acid in this case hasn't changed at all, and it's the exact same one. And so at the end of the day, even though there is a slight change to the DNA sequence, it ends up producing the same protein and amino acid sequence anyways. So overall, there's no real effect effect here. We won't notice this type of mutation, hence why it's called a silent mutation. There's a change, but it doesn't actually do anything. Okay? I mean, it is carried on, and so in the future, I mean, it potentially could, depending if this gets affected again. But realistically, we have the exact same amino acid sequence, therefore we have the same proteins being produced as a result. No harm is going to be done here. The next point, the next type, I should say, is called a nonsense mutation. Okay, and what happens in a nonsense mutation is that the protein becomes too short. The translation process gets cut off far too early because there are certain codons which code for what's called as a stop codon. Okay, and stop codons basically signal the ribosome to stop translation and finish up processing the amino acid sequence into a protein. So once a ribosome it's reading through the mRNA strand, once it reaches a stop codon, it is going to stop adding more amino acids, and it just breaks off whatever is there at that point, and then that goes off to be a protein and does whatever its job it is. Okay, in the case of a nonsense mutation, we mutated into a stop codon, and so the DNA sequence or the mRNA sequence is not read to completion. It stopped way earlier, and so there's not enough amino acids to actually build the protein. Okay, so we'll take a look at an example here. So here's our DNA sequence, our mRNA here, AUG, blah, 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 blah. Okay, this is normally what we would get if we were to read it to completion when the ribosome is going through translation. Okay, and so you get a methionine, lysine, PHE is phenylalanine, I think. Where is it at? Yeah, phenylalanine. And then we get a glycine, then we stop, and that's the amino acid sequence. That's our protein. Okay. In the case of a nonsense mutation, one of the codons is mutated into a stop codon, UAG. So if we look at UAG here, okay, it codes for a stop codon. There's a couple other ones. If you actually take a look at the chart, there are 3 in total, I believe. So when it reaches that, the ribosome is going to see that and say, okay, my job is done. Let's stop translation, and this protein that we have, this amino acid sequence we have, is done. Okay, so if you look in the original sequence, we had an AAG, which codes for a lysine amino acid. But then in the nonsense mutation, the first A there, first A got changed into a U. So now it's a stop codon. So it's reading through, instead of going all the way to completion, it does the first amino acid, and it gets to a stop, and then it says, okay, my job is done, and it's over. Okay, so that means that protein is not being made at all. That's no good to the body anymore if it's missing too many amino acids. Okay. So the third type of point mutation is called a missense mutation. And so what happens here is that the sequence is changed in a way that you get a different amino acid. Okay? As I mentioned before, proteins are quite sensitive to their shape. Okay? The shape often determines their functionality. And so if the shape is altered, they can't properly carry out their function. Okay? And so if we have a different amino acid placed in there that's not the right part, we may not actually be able to function the same way. I mean, if you think about if you're building stuff like something from IKEA, say they gave you the wrong piece. It's happened to me before. And so it doesn't fit into the place that it is meant to. And so you can't actually finish it. And so we have a useless piece of furniture now. We can't actually complete it. Or if you're building a Lego thing, if you don't have the proper piece to connect everything, then unfortunately it doesn't work the exact same way. Okay, you may be able to replace it with something else to kind of, as a substitute, but it may not fit the exact same way, and so it won't be quite as smooth, and it won't work quite as well. Okay, or it may not just work at all. So in terms of a missense mutation, okay, we'll look at this here. We have our sequence in our codon here, GGC, codes for what we know as glycine, which is up here. Use a different color. GGC makes glycine. Okay. In our nonsense mutation, that G, the first one, right here, gets changed into an A. Okay. And so when that codon is being read, we have AGC. So we get a serine instead. Okay. This again is then we get that sequence instead. It's hard exactly to say what the overall effect will be. Sometimes it can be quite bad. Sometimes it's okay. So this one, there's a little bit of a gray area. Sometimes the results can vary depending on how badly the shape gets changed. Like I said, though, proteins are often quite sensitive to their overall shape. And so if there's any changes, it oftentimes won't function as intended. It may not be quite as effective. It could just not work flat out. And in rare cases, it might actually still be kind of okay, but very unlikely so that does happen. Okay, so if we have that, we have a change in our shape, and therefore we don't function as intended. Okay, so just an overview of the three types of point mutations. Okay, so there's the silent, there is nonsense, and there is missense. Silent indicates that the change hasn't affected the overall protein shape because the codon that we got instead still codes for the exact same amino acid. So just looking at here, say we have TCC as our initial codon. In a silent mutation, okay, we change to, say, a TTT, for example. But at the end of the day, the codon, these two here, okay, they still both code for a lysine, and that's okay. And so in there, therefore, this is a silent mutation. No overall change to the protein, still functions just the same, even though it has a different base that it's reading. Okay? In the example of a nonsense, okay, we change one of the bases into a U, and therefore we now have a stop codon. So that means it's going to stop translation much earlier than it should, and we don't get any amino acid whatsoever. It's just done. Okay. Again, that's going to not be useful for us anymore. So that is called a nonsense mutation because the protein that you make from it is utter nonsense and it's useless to everybody. The last one is a missense mutation, and so one of the codons, let's say that one there, gets changed into something else. That codes for a different amino acid altogether. We get an arginine instead, and therefore the function is changed. Now, like I mentioned, the results can vary based upon what amino acids you get. So if you notice here, it says conservative and non-conservative. Don't worry too much about that. Again, if you take more genetics, you'll learn more about those. Plus, once you learn more about the amino acids themselves, there's 20 of them, in their actual structure, you'll kind of see what the differences would be in the overall effect. But a non-conservative tends to be a much bigger change from the original than a conservative one. It's still pretty close to the original structure, and so there might not be as big of an effect, whereas non-conservative, it's totally different, totally functions in a different way, and totally structured in a different way, and so we tend to see a bigger problem there. Okay. The last type of mutation is called a frame shift, and these tend to be a little bit more dangerous because an entire nucleotide gets added in or is completely removed. It's no longer replaced. Okay, we accidentally added one too many, or we just didn't add enough. Okay, and so you can think of it that way then, is that if you had, let's say, a sequence of A, U, G, 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 C, 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 just for simplicity's sake. And let's just say for some reason this original A gets deleted. Okay, that means the new sequence is going to be A, U, G, 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 C, C, C. So for one, if you look, this one isn't even a complete codon anymore, so that's not going to do anything. But also, now instead of reading all of these guys, we're reading, it's shifted the entire reading frame. And so everything after this mutation is now going to be affected. There's going to be a different amino acid for all of these guys at the end. Anything after that point is totally going to be different. I mean, you might get the odd silent one here and there that goes for the same thing, but realistically, everything after that point gets shifted down, and so there's a big effect there. Oops, there's a little bit more writing there. So either way, we're changing the order of the codons, and so the ribosome is going to read everything wrong after that point. Okay, all the amino acids after there will be totally different. Again, there may be the odd silent one, but the majority of them will be off. Okay, so we call it frame shift because the reading frame, what we're reading from, has been affected. Okay, so an example again, just looking at this one here, we have right here we got AAA. In our frame shift, we lost one of those A's. Okay, and so now everything after this lysine here is going to be different. So if you look at right here, where it was normal, we had two different amino acids here, phenylalanine, glycine, and then in the frame shift mutation, because everything has been shifted down, we get leucine, alanine, which are different, plus we don't even have a stop codon anymore. Okay, remember, a stop codon is what ends the sequence, and that tells the ribosome to stop, so it doesn't actually know what to do anymore. Okay, so that's it for the different types of mutations. Like I said, again, just to summarize, there's two kinds. There's a point mutation, and then there's a frame shift. Point mutations indicate when one base pair has been altered and changed into a different kind. And so with that, we have a silent, where there's no overall change in the protein itself. There's nonsense, where we get a stop mutation or stop codon too soon, and then translation ends before it should. And then finally, you have the missense, which means one amino acid has been replaced with a different kind, and this one can vary a little bit more depending upon which one is substituted in. Okay, we get the wrong one. Again, proteins very sensitive to their shape, so it's hard to say exactly. Frame shift, we have deleted or removed an entire base, and so that changes the frame that the ribosome is reading, and so everything after that mutation is going to be changed, likely, into a different amino acid, which then very, very greatly changes the shape. Okay, so that's everything there. Please let me know if you have any questions on that. Feel free to contact me, and I hope this makes sense. See you later. Thanks for watching, I guess. Maybe.